Hello everyone, I hope you're enjoying the conference so far. My name is Wend Yam Davy Ouédraogo, and I'm a PhD student at the Laboratory of Computational Biology in the Department of Computer Science of University of Sherbrooke. I'm working on comparative genomics and phylogeny. I would like to present you today TranscriptDB, a transcript database developed in my laboratory to study transcript conservation and evolution within gene trees. The interest of ontology between genes has led to develop several genomic databases. They provide data and methods on gene evolution. We can retrieve the information about the different types of relationships between genes, like ontology, paralogy, and gene tree. Otherwise, they do not provide information on transcript evolution, such as splicing ontology or transcript tree. So why do we need a transcription database? Eukaryotic genes can produce several distinct alternative transcripts with different functions due to alternative splicing. Alternative splicing is a mechanism that makes the transcript a refined level of functional homology relationships. More and more data on transcripts are available in open source databases, and the information on their evolution can be useful to reconstruct accurate gene trees. Different transcripts from one gene may have different functions and may be localized in different tissues. More knowledge about transcripts would help to better understand the evolution and functions of genes. So we propose TranscriptDB, a database and analysis tools developed in our lab. TranscriptDB is a transcript-centric database to study transcript conservation and evolution. It implements a new method to compute clusters of conserved transcripts in homologous genes. What is the TranscriptDB content? TranscriptDB contains data that were obtained from ENSEMBLE resource. The data for three different versions of ENSEMBLE, respectively, the version 97, 98, and 102 are included. The figure shows the number of genes, the number of transcripts, the number of species, the number of gene trees retrieved from versions of ENSEMBLE. We can easily notice the increasing amount of transcript data shown in green here. TranscriptDB provides type of homology relations between transcripts, also transcript trees and gene trees. It also provides comparative genomics data display, tools for data analysis and processing. It also provides data export and API. TranscriptDB allows you to download data in FASTA format or CSV format. The collected and computed data are loaded into a custom PostgreSQL relational database. The database provides relevant clusters of conserved transcripts and transcript phylogenies computed thanks to a new similarity measure. So the TranscriptDB clustering algorithm is a graph clustering algorithm designed to identify conserved transcripts between homologous genes. It takes at the input a set of transcripts of homologous genes and gives clusters of conserved transcripts at the output. The algorithm relies on two steps. The first step is a pairwise transcript similarity measure, and the second step is the reciprocal best hit clustering. The first step, the transcript similarity measure, consists in first, compute a multiple spliced alignment of transcripts given at the input. We first align gene by using a tool for computing multiple sequences alignment, and after that we retrieve the alignment of transcripts by using the alignment of gene sequences. Then we compute the transcript similarity score for each pair A and B of transcripts, such as A and B are not produced by the same gene. The transcript similarity score accounts for the quantity of nucleotides shared, called nuc-om, which is equal to the division of two terms. The first term here is the summation of matches or mismatches, and the second term here is the present result, is the sum of the precedent results and the total number of indels. In this example, we have 5 indels and 14 matches or mismatches. so the nuke-ohm score is equal to 0.73. The transcript similarity score also accounts for the quantity of exons shared called exon ohm, which is equal to exons blocks in common divided by the set of all exons blocks. In this example, the number of exons blocks shared is 2, and there are 4 exons, so the exon ohm is equal to 0.5. The colored rectangle in this alignment represents the exons blocks. Finally, the transcript similarity score is equal to the half of the sum of nuke-ohm and exon ohm. So the transcript similarity score of this example is equal to 0.62. So the second step, the reciprocal-based clustering, consists to first compute the symmetrical score matrix which resumes all the pairwise transcript similarity scores. Then we construct a graph homology based on the score matrix where the nodes represent transcripts and edges have weights equal to the corresponding score in the precedent matrix. Finally, we conserve transcripts that share the best score together. There is also an algorithm for transcript phylogenesis reconstruction developed in our lab. The algorithm takes at the input the clusters of conserved transcripts from TranscriptDB clustering and the corresponding gene tree and give at the output a transcript tree. The algorithm has 2 steps. The first step is the transcript subtrees construction and the second step is the transcript supertree construction. The first step constructs the transcript subtrees in congruence with gene trees for each transcript cluster resulting from the TranscriptDB clustering. Then, the second step is to construct a transcript supertree by combining all subtrees based on the transcript similarity measure and reconciliation with gene tree. The optimization criteria is to minimize the number of creation in the resulting transcript tree. The resulting supertree is used to infer homology relationships between transcripts. Here we have the overall view of the method proposed that combine the TranscriptDB clustering algorithm and the transcript phylogenesis reconstruction algorithm. So what type of data can you get in TranscriptDB? We can get transcript level resources like clusters of conserved transcripts, transcript trees, and stable ID mapping for transcript trees, the types of homology relationships between transcripts, and also the decomposition in exons blocks and the conservation scores. Also, we can get gene level resources like gene trees, gene splicing structure, and the set of exons. We can get also the types of homology relationships between genes and the alignment of homologous genes. So the TranscriptDB web interface procures a quick access to transcript information. Here we have a human transcript given by a user. We have automatically access to information about clusters of transcripts, the information of corresponding gene like stable ID of gene, stable ID of gene tree, stable ID of transcript tree, the biotype, the strand of gene, the number of exons present in the transcript. TranscriptDB provides also an interactive view of the exon entrance structure of transcripts by highlighting the exons. The data can be downloaded by the user. The web interface gives a full perspective of the transcript sequence that gives a unique color to each exon to assure a comprehensive exploration of the structure of transcripts. It also shows a table that describes the conserved transcripts and the score associated with the alignment of the decompositions in blocks, shown here in red. Here, there is an example of a transcript cluster resulting from TranscriptDB clustering. So in addition to provide an interactive view of gene tree, TranscriptDB provides a multi-scale graphical view to highlight conserved transcripts into the corresponding gene tree. Then we can get gene-based resources in TranscriptDB. Indeed, we can retrieve gene tree and all information about homology between genes, like I mentioned in my precedent slide. We can see here the exon-intron composition of all transcripts produced by a given gene, and TranscriptDB highlights the conserved exonic regions in red here. Also, the data can be exported. We have the description here of types of homology relationships between genes, the transcript table that shows the location of transcripts into the gene, and we have visualization and comprehensive tools that offer you a description of data, like here we have a description of transcripts of homologous genes. To conclude, we presented a transcript-centric database and a new method based on splicing structure to compute clusters of conserved transcripts to the reconstruction of transcript and gene phylogenies. The database provides tools for analysis and visualization of genomic data. The database content is accessible through a REST API, and the tools is freely available through its web interface at www.transcriptdb.copius.usherbrook.ca. I would like to thank the Canada researcher, the University of Sherbrooke, and my supervisor and all the members of Copius Laboratory. It was a pleasure to present our research to ISMB 2022. Thank you.